apologise for my voice first. I've been at London Tech Week, and between that event and this event, I've absolutely thrashed it. So I might have to cough in the middle of the talk. But thank you very much for having me. It's a real privilege to be amongst. You think do this one?
You can multimodal, right? You can
Hello. To be featured amongst such great talks that we've seen yesterday and today. I'm Jake. I'm a bioinformatician and machine learning scientist from Twig. We are a biomanufacturing company for the fine chemicals and cosmetics industries. In practice, that means we leverage some of those well-studied microbes which are abundant in metabolic engineering to produce high-value chemicals for things like skincare, fragrances. Today, I'm going to talk about one of our AI initiatives, Canopy, which is a Heterograph Foundation Model for metabolic engineering, and how we built the underlying knowledge graph, which backs this up. This is kind of a systems paper that we're putting out in a few weeks, so keep an eye out for that if you are interested. Just a quick motivation framing. Previously at Twig, we had lots of different independent models that had single uses. So, for example, they predicted the ideal gene order of a synthetic pathway, or they told us which knockouts to perform for optimizing titer of these high-value chemicals. Canopy was, in one framing, was a way of sort of amalgamating all of those and replacing them with with reusable lightweight probes trained on knowledge graph embeddings. So, as I said, Canopy is a knowledge graph and foundation model for strain design. We assemble the graph using multimodal data from public biological databases. We mine literature fermentation experiments, and I was grateful for Henry earlier to introduce the concept of DBTL, because that's something EngBio people love to talk about. But we also include our own design, build, test, learn strain engineering experiments from Benchling, our lab information management system. We have approximately 7 million nodes, 11 million edges, and these are across 13 different node types, from stuff like genomics, proteomics, and these experiments that I described a moment ago. 3 modalities are given a special attention. So we use frozen foundation models to embed them into an embedding space, which we then feed into the graph. And so these are protein sequences. We use ESM, chemical structures, and free text, so descriptions of the experiments, of the strain names, and other sort of abstract node types. In terms of the long-term framing for this, we thought that this sort of engineering biology maps quite nicely onto a knowledge graph, as you'll see in a moment, and we use it across biological scales, from sort of real-life tangible things like the temperature of your biofermenter, all the way down to sort of single DNA mutations which define individual organisms. Another motivation for this work was to incorporate as much data from our laboratory and partner laboratories, and we're expanding this into sort of new modes as well, like images of cell colonies and cultures and Raman spectroscopy. And then in the long term, we're trying to scale this beyond towards frontier scale foundation models, use lab-in-the-loop reinforcement learning. And also one of our interesting ideas is to bring in our other AI sort of topic at Twig, which is multi-agent research systems. And we'd like to perform some reasoning over highly attended nodes when we make predictions using the graph. So this is the schema of the Canopy knowledge graph. As you can see, there's sort of 5 classes of data from the different omics. The core node type in our work is definitely the strain, as this acts as a hub joining the taxonomic tree with genetic modifications, with the heterologous pathway that we're deriving our compounds from, and to linking to the experiments where we keep all the conditions that they've been experimented under. In this graph, you can see they all have directed edges, which means you can read the sort of the semantic triplets of the knowledge graph quite well, such as gene encodes UniRef cluster, which is described by a gene ontology term. But actually at training time, we add reverse edges as well to allow knowledge passing through the graph bidirectionally. As I said earlier, this is about 7 million nodes, so this is probably a little bit too big to fit on even like a H200 GPU. So we perform a lot of graph sampling and use MetaPath and BFS expansion to pass reasonable subgraphs to our downstream training models. One data type is perhaps slightly overrepresented, which is the transcriptomics. This is because there's two very, very valuable data sets for understanding gene expression in E. coli and Saccharomyces, which are like these two workhorse organisms for metabolic engineering. So that represents about, 5 million nodes in total, but that's tells you the level of all of these different expression of genes under many, many different conditions. Okay, so this is the schematic of the model we're working on. As you can see on the left, we take in 3 different input data streams. This is the node data, which goes through a simple linear embedder into model dimension space. We use spectral positional encoding on the whole graph so that we learn global graph topology, which goes through a sign invariant network also into the model space. And then on individual graph samples, we encode local structure using random walks. And so these are all summed in the input. At the top, you can see we use a relatively standard heterograph transformer layer in our model. This is similar to other multimodal attention networks that we've seen the last couple of days, but it's from, we have a layer per source destination edge type. We do some neat sort of engineering to incorporate features from frontier language modeling into these sort of graph formulated models. So this is stuff like the scaled residuals for both the attention and the feed-forward network. So these are learned residuals, so they give different importance depending on how the model weights are formulated. We also do something called jumping knowledge aggregation, which is a max pooling step where we cache the output of each individual layer, and we do max pooling across all of those. And that is to preserve short-range interactions so they don't get over-smoothed by the long-range interactions as we add more layers to the model. The final thing that I wanted to highlight here is that we also add virtual nodes during training. So these are an extra node type which have bidirectional edges to every other node, and that allows long-distance message passing through the graph during training. We remove them during inference. As you can see at the bottom, so we ablate these against a downstream task, which is tighter prediction. We get a reasonable R-squared with the full model, and the virtual nodes in particular are really useful. Those long-distance interactions through the graph are essential for downstream application performance. Frequently with these multimodal models, you have to do quite a bit of objective engineering to get them to do exactly what you'd like. So we use 2 relatively standard graph completion objectives. That's the link prediction, predict whether or not 2 nodes are connected, and the graph distance objective, which is how far apart are a pair of nodes. But we also add, we thought we'd be doing lots of downstream regression using these models, so we also add a regression objective across all node data types. And perhaps with the most sort of opinionated choice here is to add an experiment contrastive objective. So we define the objective embedding, the experiment embeddings as best as possible. And once again, we ablate against this downstream tighter prediction task, and that contrastive learning loss is really important for performance, as you might expect, because we are using those experimental embeddings. So just to wrap up, we've assembled a really large, nice synthetic biology knowledge graph. We use 3 sort of critical modalities with these frozen embedders. Our heterograph transformer beats tabular baselines by sort of 3 times. And that there's a sort of a humble improvement in performance as we scale from 500 million to 3 billion parameters. And that's using 5-fold cross-validation, so it's a pretty rigorous evaluation. As I highlighted, we did some ablations which isolate which things are important in terms of our systems engineering. And then sort of with the aim of focusing on deployment, and one of UKO main's key focuses, we're already validating this in the lab, proving out our foundation model claim. So we're doing a number of experiments to predict beneficial knockouts for manufacturing, for ordering the genes in our expression plasmids, and for completing the incomplete metabolic graph, which forms part of the heterograph. We're also looking to scale the data and the parameters, and established scaling laws for this model type. We're currently working quite hard to add protein structures. I wrote a nice PR for doing this week, and my boss Sat has added sort of 100 million metagenomic protein sequences. So the graph is growing quite quickly, and I'm hoping the modeling can keep up. But yeah, if you have any questions about this, if you want to talk about BioML, please come and meet me at lunch. Thank you very much for hosting me, and to Air, Sov AI, Dawn, and Isambard for continued compute support. Thank you.
2 quick questions. Yes, please.
Thank you very much. It's fascinating. So I work in an area which is called Neurosymbolic AI, and obviously it's working with fusing generative AI, foundational models, neural models with knowledge bases, graph structured, etc. But I was quite curious that you not once mentioned the word neurosymbolic. So I was just actually wondering, are you guys sitting in this box, or are you not quite there, or you outside already and left neurosymbolic behind?
Yeah, I think it's an option we considered, but it was sort of beyond what we'd worked on previously in our expertise. I built a similar system to this for metabolic engineering previously in another role at another company. So we were working on expanding that as a specific project, but it's definitely something we've thought about but not actioned yet.
Great. Yeah, if we have more questions, maybe we can talk with Jake during the lunch break. Great. Yes, let's thank Jake again.